As a biomedical engineer, I've been using ASAP since 2005, and I've applied ASAP to several bio-optical systems such as tissue modeling, sensing applications such as blood oxygenation sensors, and imaging systems like confocal microscopes. We actually have the capability of coupling a real optical system, as far as a virtual model is concerned, with tissues that have realistic optical properties, so that when you trace through, you get an accurate result based on what is generally the most important part of the bio-optical system, and that is the scatter and absorption that you get from tissue, which can be more complex than the system that you've built to analyze the tissue. Now I'll provide an overview of the realistic skin model in ASAP, the tissue generator in ASAP, as well as applications such as modeling fluorescence in bio-optical systems, analyzing treatment efficacies in situations such as photocoagulation, and the power of ASAP to import and utilize geometries seen in real-world bio-optical systems. One of the most powerful and useful features of ASAP is the ability to combine accurate tissue optics with realistic biogeometry characterizations. Let's look at a few such examples. This bladder model that you see was created by the Breault School of Medicine at East Carolina University from CT scans of an actual patient. These CT scans were then reconstructed into a three-dimensional model, which we can import into ASAP, apply optical properties to, and trace rays through in order to get an idea of the light distribution within the wall of the bladder. In order to more adequately visualize this, let's take a look at this in 3D. We can remove portions of the bladder in order to examine the scatter and absorption within the wall of the bladder. As you can see, we have an isotropic point source that is propagating through a saline solution inside the bladder, and scattering is occurring within the wall of the bladder. And we can visualize this using our voxel system. We can actually see absorption in the medial, sagittal, and transverse planes. We can visualize this as well as a single slice through the center of the bladder. This is important for applications such as photodynamic therapy, where we would be creating a probe that was placed in the bladder and illuminating the interior of the bladder in order to activate a photosensitizer. In a lot of these applications in the past, there have been problems with creation of fibrosis or scarring in the inside of the bladder, basically turning a non-invasive procedure into an invasive procedure later on when they have to remove the scarring. What we can do is we can examine the light distribution on the inside of the bladder and compare that to previous simulations where only a spherical bladder was used as an approximation, and determine, based on the structure of the bladder, what the ideal probe shape is in order to get uniform illumination on the interior of the bladder. Now let's take a look at a few other examples of how geometry has influenced design in bio-optics using ASAP. This rat brain model, which was created based off of an MRI slice of the rat brain, was used by Stanford to characterize a system to examine activation of specific regions of the brain by examining blood oxygenation concentrations, so that we can create a non-invasive method of examining the activation of specific areas of the brain without having to use devices such as MRI. This chest wall example was used by the Breault School of Medicine to examine photodynamic therapy treatment in a post-mastectomy patient. They've used an external LED-based source and are illuminating the chest wall in order to activate a photosensitizer and remove any potential cancerous cells that are left over after the mastectomy. Complex biological structures are not the only structures that may be interesting in bio-optics. For instance, this complex tracheal mesh is used in patients that have tracheal or esophageal cancers in order to hold the esophagus or trachea open and facilitate breathing or swallowing. Now, this mesh can become intertwined with the tissue of the trachea or esophagus and requires photodynamic therapy treatment in order to remove excess tissue and reopen the passage. As you can see, this is an incredibly complex mesh design, and therefore it's important to be able to model each individual portion of this mesh to get an accurate depiction of the optics that are necessary in order to effectively treat this situation. Finally, we can see here a finger model with realistic optical properties for stratum corneum, epidermis, dermis, musculature, and bone. And this model is integrated with a blood oxygenation sensor, including a parabolic mirror, an LED, and underneath a detector, so that we can examine the level of blood oxygenation within the dermal layer of this particular model. This is a great example of how we can create a complete virtual prototype using both a biologically relevant structure as well as realistic optics. And these are just a few examples of how tissue models can be applied to complex biological structures in ASAP to help us better understand complex optical systems for bio-optics. So we can use the tissue modeling capabilities of ASAP to model different disorder and disease states, and also to model certain treatment types, such as photocoagulation. So what we can see here is we have a skin model with stratum corneum, epidermis, and a dermal layer, and it is also filled with blood vessels. And the rays propagate through and are depositing energy within the volume. You can hide those rays and take a look at the energy distribution. And this is at 350 nanometers wavelength. And we can see that we have a fairly large spot on the layer of the blood vessels where energy is being absorbed outside of the blood vessels. We can also hide the blood vessel wall and see that we have relatively high energy absorption in comparison to the energy absorbed outside of the blood vessels at this layer. Now, if we grab this layer and we drag it up into the epidermis, we can see an even higher energy absorption here. We can compare that to the same basic geometry, but with different optical properties. These optical properties are at 560 nanometer wavelength. We can hide all of our layers once again and only take a look at the rays that are scattering in the system. As we can see, there's a large number of rays that have actually penetrated within the dermis and have scattered around, versus the previous model, which we could only see a very few number of rays within the dermal layer. If we take a look at the same sort of energy profile in this 560 nanometer trace within the blood vessels, we can see, hide these blood vessel walls, we can see energy absorbed within the blood vessel wall, and also a lower amount of energy absorbed in the epidermis overall. We have different ways of viewing this as far as display properties. We can go to an isometric view, and this is a very clear view of how our energy is absorbed in the system. You can see a small amount of absorption in the stratum corneum, and then a huge spike in absorption in the epidermis, and then a fall off. And within the dermal layer, we can see very small spikes where energy is actually reaching the blood vessels. Comparing that to 560 nanometers, we can see a relatively small amount of absorption in the epidermis, still some epidermal absorption, but then in comparison, a huge amount of absorption in the blood vessel. So as far as treatment efficacy, 560 nanometer wavelength light is much better at dealing with photocoagulation than 350 nanometer light, because you're going to have collateral damage as far as the skin is concerned here. So let's take a look at how we would accomplish tracing these particular examples within ASAP. The first example we'll look at is the 350 nanometer blood vessel model. We've got stratum corneum, the epidermis, and the dermal layer, as well as the blood vessels. This is a model that was created using the realistic skin model in ASAP, and therefore it's a wavelength dependent model. Each of the tissues that we see has specific absorption and scattering characteristics that are unique to that particular wavelength, in this case 350 nanometers. What we can see is we can see a lot of rays that aren't penetrating within the dermis. They're actually stopping in the epidermal layer. And a small amount of rays that are actually getting to the dermis are getting into the blood vessels and basically dying in the blood vessels, being completely absorbed. Now, this is a qualitative analysis as far as where our light is going. We can look at a more quantitative analysis, which is our voxel system and the isometric graphs and picture graphs that we have available. As we can see, and we've seen before, we get a very high absorption in the epidermis at 350 nanometers wavelength. And very, very faintly, we can see the blood vessels within the dermal layer here. But the energy that's absorbed in those blood vessels at 350 nanometers is very small in comparison to the high absorption in the epidermis. Once again, we can see in the isometric view of this particular trace, high energy absorption in the epidermis, low energy absorption in the blood vessels. Now, comparing that with the same skin tissue phantom at 560 nanometers, we have the same setup: stratum corneum, epidermis, dermal layer, blood vessels, the same geometry, but the optical properties have changed due to the fact that we've changed the wavelength of the light that we're looking at. So light will propagate through this system very differently because we're looking at 560 nanometer wavelength light versus 350 nanometer wavelength light. And we can see a much larger number of rays propagating into the dermis, getting to the blood vessels, and also scattering around within the dermis and getting to blood vessels that are outside of the range of our source, which may be incredibly useful for treatment applications. Looking at a pictorial representation of that, we've got relatively low absorption in the epidermal layer, and we can see a profile here of the low absorption in the epidermis and then high absorption in the blood vessel wall. And if we return to our isometric view, that's also reflected here as low absorption in the epidermis, high absorption in the blood vessel. So to summarize, we can use skin models and other tissue phantoms within ASAP to examine treatment efficacies based on wavelength so that we can optimize optical systems in order to better serve the patient as far as creating an optical device. ASAP has the capability to model fluorescence in optical systems. Coupling this capability with scatter and absorption in tissue phantoms provides a useful feature set to help examine bio-optical devices such as fluorescence microscopes. In this example, we've used the realistic skin model to create a tissue phantom, and then placed within that tissue phantom a fluorescing body, which we can excite and then use the energy from to create a new source for re-emission. And what we see is a skin model composed of the stratum corneum, the epidermis, and a dermal layer. The dermal layer includes our fluorescing body. The blue rays that you see here are actually our excitation rays, and that excitation is around 515 nanometers, which is the excitation for yellow fluorescent protein, which we've modeled here. The yellow rays that you see are actually the re-emission rays from the yellow fluorescent protein, and as you can see, they've scattered within the volume. So we're actually modeling the real effects of scatter and absorption of the dermal, epidermal, and stratum corneum layers, so that when you take a look at the distribution of light as it comes out of the system, you can compare that with the volume that you've created within the system to see how it differs as far as size and shape. And as we can see, there's a very marked difference in the size of the distribution of energy on our detector versus the size of this particular yellow fluorescent protein body. Now let's take a look at how we actually accomplish this. First, let's run this file and take a look at how we deposit energy within the skin. Now what you see in the blue here is the excitation source at 515 nanometers, and the yellow sphere within the dermal layer is our yellow fluorescent protein body. And we're depositing energy in such a way that we can track the energy within that volume and then use that energy to create a new source. Basically, what we'll do is turn the source inside out and re-emit the source, and the energy is going to be scaled based on the quantum efficiency, so how well the fluorophore can actually re-emit that energy, which in this case is about 61% for yellow fluorescent protein. And as we see here, we have a distribution on a detector that we've created just above the surface of the skin, and it's a very, very diffuse sort of energy distribution, which is what we would expect from scattering and absorption from re-emission of a source within another scatterer and absorber. What we can do with this is we can actually use this to examine optical systems that would work with the skin, such as fluorescence microscopes, so that we can optimize or idealize our optical system to get the best image quality out. So to summarize, what we've accomplished is creating an excitation source which has deposited energy within a yellow fluorescent protein body. We have taken that energy and re-emitted it as fluorescence. And the fluorescence is scattered and absorbed within the skin in a realistic manner. And we have collected on the surface of our sample the energy as a distribution that can be used in a very realistic manner to examine fluorescence systems in skin tissue. With the tissue generator, ASAP possesses the capability to characterize any type of tissue based on user-defined parameters. These tissue models could be useful to model optical systems for extracting optical properties from tissue samples in laboratory settings. In this model, we have a 0.2 millimeter slab of tissue, which is illuminated with a 630 nanometer light source. We can examine the scatter as it passes through this particular slab of tissue, as well as the absorption within the tissue using a voxel system in all three dimensions. And we can examine a detector which collects the light that has passed through the tissue and it has been attenuated. So let's take a look at how we could create such an optical system. Imagine we have an INR script, an ASAP script that contains a detector, a Gaussian source, and some plot, trace, and analysis information. What we're really missing here is a tissue model. In this case, a 0.2 millimeter slab of human liver tissue. Using our tissue generator, we can quickly and easily create such a tissue. What the tissue model builder requires from us is the absorption coefficient, scatter coefficient, anisotropic scatter coefficient, and thickness for each of the layers. Now here we're going to use centimeters as our unit of length for our geometry as well as for our optical coefficients. Use 630 nanometer wavelength light. Use a surface area of 10 square centimeters. A single layer. We'll use Fresnel effects, and we'll give the name test liver to our file so that we can easily find it later. And here we need to input the optical properties of our tissue, as well as the layer thickness. So we need a 0.2 millimeter thickness for our sample, and we need an anisotropic scatter coefficient, absorption coefficient, and scatter coefficient for human liver. Now, these optical properties can be found in published literature. For instance, for 630 nanometer light, we can find human liver optical properties in a review of the optical properties of biological tissues, which is published online in PDF format and provides a wealth of information about different types of tissues for different animals. We can see that we have an absorption coefficient, a scatter coefficient, and a G value for human liver at this particular wavelength. And those correspond very well to our anisotropic scatter coefficient, absorption coefficient, and scatter coefficient in the slab tissue model builder. So let's input those parameters. Got 0.95 for our G value, or anisotropic scatter coefficient, 3.2 for our absorption coefficient, and 414 for our scatter coefficient in inverse centimeters. We are not provided with an index of refraction for the human liver in this particular case, so we'll assume a value of 1.45. At this point, the tissue generator has created a file that includes not only the optical properties, but also the geometry for our slab tissue. What we can do is go to our working directory, find our file testliver.inr, and extract the information from the file by copying and pasting into the optical system that we've already created. And now we have a complete optical system, including not only our source and detector, but also the slab tissue geometry and optical properties. Let's run this file and examine the output onto a detector plane as we saw previously. We can see scatter occurring within the tissue, which is causing spreading of our source on the detector. And that's obvious from the fact that we began with a 0.25 millimeter radius source, and we have spreading as far out as around 0.5 millimeters. Returning to our 3D viewer mode, we can see we start out with a much smaller size detector, and as the light passes through the liver tissue, it spreads out to a much larger size. So to conclude, using the tissue generator, we can create slab tissue models of one or several layers that will allow us to characterize optical systems in a very realistic and helpful way in order to better understand the way that our systems work. The Realistic Skin Model, or RSM, in ASAP is a useful tool for generating biologically accurate optical tissue models of human skin. What sets the RSM in ASAP apart from other skin models in optical software is the ability for the user to specify source wavelength with a range of 250 to 1,000 nanometers, and the ability to manipulate optically relevant molecular concentrations to model disease states in skin models. Now, what we see here is an example of such a skin model. We have a 3-layer model, the stratum corneum, the epidermis, and a bulk dermal layer. And we can see as a 560 nanometer wavelength source is traced into the system, the scatter properties of that system. Now, if we take a look at this same trace in a 2-dimensional plot, what we can examine is the specular and diffuse reflectance from this trace, and we can see that several rays have not only directly reflected off of the surface of the skin, but also entered the skin, scattered, and then escaped back through the surface. And this information can be relevant in sensing applications and imaging applications. If we return to our 3-dimensional model, then we can take a look at how we can track the energy in this model using a voxel system. And this is fluence tracked as a function of all 3 dimensions in the same trace, 560 nanometer wavelength source. Now let's compare this to a same trace with tracking absorption in the skin. And we can see that there's a marked difference between the energy absorbed in the skin and the amount of energy flowing through the skin, or the fluence in the skin. Other ways to visualize such energy are provided in ASAP through plots such as an isometric plot. And this is an isometric plot of the fluence that we were just examining. And you can see a very gradual fall off in energy. Similarly, in a contour plot, we can look at fluence as a function of depth, and we can see deep penetration within the skin for energy. Now, comparing those to the absorption plots, we can see a high confinement of energy in the epidermis, and then a steep drop-off and fairly lower absorption in the dermal layer. And this is reflected in the contour plot. In fact, so much so that the energy that's absorbed within the dermis is beyond the bounds of our scale for this particular contour plot. Again, looking at the same information in a different manner, we can visualize the fluence in the skin in a pictorial representation and examine in the Z and X dimensions the fluence throughout the skin model. We can see a gradual decline in energy and a gradual, or a rather steep drop off on either edge, but a gradual rounding of the source as it propagates through the system. Now, comparing this once again with absorption, we can see a dramatic drop-off as we reach the dermal and epidermal junction, and then rather low absorption throughout the dermis, with a similar profile in the X dimension. Now let's examine how we would model this example file using the RSM dialogues. The first dialog that we see is a realistic skin model initialization dialog, where we can specify options such as the sample surface area and the wavelength. The wavelength is incredibly important to the realistic skin modeling system in that it allows us to set the optical properties of the skin for this particular wavelength. We will check off the Use Default Material Parameters box. If we use the default material parameters, the RSM is preloaded with variables that are taken from published literature and would give you a generalized skin model that's useful in many situations. However, in light of the fact that we are examining particular chromophore concentrations in the skin, we want to not use default parameters and instead look at specifics of the skin. We will use the default source and perform dialogue-driven analysis. The realistic skin model provides 3 methods for model creation. Visual characteristics, which, if the quantities are unknown for chromophore concentrations or specific optical properties such as absorption and scatter, then we can use the visual characteristics of the skin, the lightness or darkness of the skin, say, to qualitatively specify a skin model that may be useful in certain situations. We can also specify our skin model using chromophore concentrations, which is a direct method of controlling the exact concentration of light-absorbing molecules in the skin. Our third option, direct entry, allows us to measure skin within a lab environment and then take those measurements, transform them into scatter, absorption, and anisotropy coefficients, and then input those into the RSM to create a virtual tissue that will last even when the real tissue degrades. For this situation, let's take a look at chromophore concentrations. Now, we're provided with the ability to model multiple layers of skin: stratum corneum, epidermis, dermis, hypodermis. For our situation, we looked at the stratum corneum, the epidermis, and the dermis, so we'll use the default values for this. The RSM provides the ability to model both a single-layer bulk approximation of the dermis or a 2-layer papillary and reticular dermal model, which will allow us to look at the interdigitation between the epidermis and the papillary dermis. Our model was a single-layer bulk approximation, and so we will use the default for this as well. The RSM provides several optically relevant and physiologically relevant geometries, such as hair and blood vessel modeling, which we won't examine in this particular model. For each layer modeled, we can examine and change the thickness, the anisotropic scatter coefficient, and the index of refraction. Also, for each layer, there are confined chromophore concentrations, such as beta-carotene and water for the stratum corneum. If we change, say, the volume fraction of water in a particular layer, we can modify that layer to examine states such as dehydration. The epidermis, once again, presents us with the ability to change thickness, the anisotropic scatter coefficient, the index of refraction, as well as chromophores that are specific to the epidermis, such as eumelanin, which is a brown pigment, and pheomelanin, which is a yellowish-red pigment. And the ratio of these two chromophores controls the relative color of the skin, which allows us to model different ethnicities. We can also model, once again, beta-carotene, water, and control the total volume fraction of melanosomes, which, when increased, will darken the skin. In the dermis, we're presented, once again, with the thickness, anisotropic scatter coefficient, and index of refraction. And in the dermis, we can control the amount of hemoglobin in the blood, which, if we reduce it, can mimic some anemic states, the concentration of beta-carotene, the concentration of bilirubin, which when elevated can cause jaundice and can be optically relevant in certain situations, the volume fraction of water, the volume fraction of oxyhemoglobin, which is a ratio of oxygenated blood to deoxygenated blood. If we control this value, we can also model hypoxic states, and the total volume fraction of blood in the dermis. If we increase this volume fraction, we can model states where a high amount of blood is present in the dermis, such as a high vasculature that can be seen in things such as port wine stains. At this point, the realistic skin model has created both the geometry and the optical properties of the skin, and now we'll proceed to our source creation, trace, and analysis. Our default source will be slightly less than the number of rays that we used for the previous trace for expediency. We will randomize our rays and use a total source flux of 10 milliwatts. Our trace options menu also presents us with a wealth of different options. such as the number of pixels, the fluence or absorption tracking in our voxel system. We'll choose fluence. The window of interest, which allows us to examine the voxel system at different views from different perspectives. The area and depth to track in our voxel system. The number of slices in the dimension that we are not viewing in order to get a depth resolution. Plot options, whether or not we would like to visualize none of our geometry arrays, geometry only, or examine both geometry and rays in a full plot, as well as the ability to plot only a subsection of the rays that are being traced. We can also choose the ray color, which in no way impacts the wavelength that we've chosen previously. We will choose green, and now you can see the stratum corneum, epidermis, and dermal layers, as well as the source being traced through the system. And we can see, once again, a high amount of scatter within the system, as well as specular and diffuse reflectance from the surface of the system, and rays that have escaped through the back of the system. These are generally low energy rays and can either be neglected or collected on this dermal surface for analysis later. Now we're presented with the ability to perform analysis via a dialog system. The RSM calculates, based on the number of slices and the size of our analysis region, the spacing between the slices, which will allow us to more accurately select which slices are of importance to us. So, for instance, we will view a single slice that slices through the center of our system, slice 75, and display that single slice. We can also average our information before we display it, and that average is over 1 pixel in every dimension. We can use a logarithmic scale, which we won't do in this particular case. And we have 3 output types, which we've seen before: isometric, contour, and a picture display. Now, our 3 display types will look a bit different from the display that we had seen before due to the fact that we traced a lower number of rays. The RSM also provides a method by which we can return to the analysis in order to perform an additional analysis on the same data set. Moving forward, the realistic skin model allows us to output the code for this particular skin model to a file so that we can use it later in a separate optical system. If that optical system is well defined, we can change the position and rotation of the skin model to position that skin model within that optical system before we output it to a file in the X, Y, and Z dimensions for position as well as rotation. Finally, the RSM allows us to either output scatter models only, which would allow the user to apply these scatter models to previously defined geometries, or output both scatter models and geometry, which would create a slab tissue model similar to the one that we've seen before. We can also provide a unique file name for our skin model so that we can easily find it in our working directory. Our model output is complete, and the RSM has finished. Now let's take a look at the output that we've obtained. The name of our model was skinmodel.inr. And it's a complete working INR script that can either be used as is or copied into a separate optical system to be combined with an optical device so that you can examine both an optical system and a biologically relevant tissue phantom simultaneously. So to summarize, what we've seen here is the RSM's ability to create human skin models based on user-defined parameters, such as wavelength and optically relevant molecular concentrations, something that no other optical software tool can do